So I want to welcome Dr. Gagandeep Kang, who is one of the foremost Indian virologists. She's a physician scientist at the CMC Christian Medical College, Vellore. She was the executive director of the Translational Health Sciences and Technology Institute in Faridabad, an autonomous institute of the Department of Biotechnology, Ministry of Science and Technology. She's a leading researcher with a major research focus on viral infections in children and the testing of the rotaviral vaccine. She was awarded the prestigious Infosys Prize in Life Sciences in 2016 for her contributions to understanding the natural history of the rotavirus and other infectious diseases. In 2019, she became the first Indian woman to be elected as a Fellow of the Royal Society. Welcome, Dr. Kang. Over to you, ma'am. Ma'am, you're on mute.
Thank you. So what I'm hoping to do in the next 20 minutes is just to give you an overview of COVID-19 vaccines, variants, and myths. And that last bit was added on because Ajay asked for it. So can you see my slides?
Yes, we can. Yes, we can now.
Okay. So just to dive into where we are today, we have 5 vaccines that are in limited use, and we have about 8 vaccines that are in full use, and we have lots of vaccines that are still in development. We've never made vaccines this fast, and we've never started to use them on this kind of scale. And because of that, there is a lot of data that comes out that people don't necessarily always understand or know how to deal with. And particularly that relates to the safety of vaccines, as well as to some extent their efficacy. Now, the platforms that we have are the platforms that either deliver the message, which is the bottom, or the vectored vaccines, the RNA vaccines, and the DNA vaccine, which Zydus Cadila is developing. We also have the old style vaccines, which is the live attenuated inactivated virus-like particles and proteins that are produced through recombinant DNA technology. And the interesting thing about this virus is our very, very heavy dependence on one protein of the virus, the spike protein that everybody has heard about. But practically all vaccines, other than the inactivated and the live attenuated vaccines that are in development, all of them depend on the spike protein. And that makes sense because the spike is really what allows the virus to attach to the host cell and get inside. Now, the protein itself is a somewhat floppy protein, and it has been possible through work that was done at the National Institutes of Health to make that protein a little bit stiffer, which allows us to generate a better immune response to the virus. Now, when we started out to develop these vaccines, we actually based a lot of our thinking on influenza, and we had hoped to have a vaccine that was at least 50% efficacious, that didn't need more than 2 doses, that was something that would be usable in adults and would give protection for at least 6 months. Where we are today is every vaccine for which we have efficacy data does better than the target product profile had wanted. And of the 4 vaccines that have failed, none of them failed because they did not have sufficient efficacy. Either they dropped out at the stage of immunogenicity, or it was decided not to proceed with the vaccines because there were other competitors that were further along in the pipeline. So I think one lesson that we have learned is that there are ways of making vaccines faster than we had thought was possible. And we have managed to increase the probability of success by designing vaccines based on prior knowledge. We have a lot of vaccines with efficacy data. These include the mRNA platform, the adenovirus vectored platform, lots of inactivated vaccines, and Novavax with the first protein vaccine, which has efficacy data. Now, how do you use vaccines? And this is an issue. And in order to decide how vaccines needed to be used, there was probably about 6 solid months of discussion at WHO with countries trying to decide, in a situation where we are going to have slow and limited supply, what is it that we need to be doing to ensure that we can get vaccines to as many people in the world as possible. And based on that, it was decided that the goal of vaccination was going to be to try and protect people who were going to be responsible for healthcare and protect those people who were at high risk of severe disease. So this was really healthcare workers and essential workers and high-risk adults. And it was calculated that 3% of the population would cover healthcare workers in most countries. And then if you went up to 20%, you would be covering all high-risk adults. Now, as we know, in India, we've jumped around a bit. We did go for healthcare workers and essential workers first. Then we said we were going to do over 50-year-olds. When we started with 60-year-olds, and now we've dropped it to 45-year-olds. And we're talking about whether we should be changing this approach or not, and the reasons for that change. Just to point out that some countries have decided, even countries with limited supply have decided not to target high-risk populations first, but to target those who are most likely to transmit infections, to allow for control of the pandemic. And that's not a choice that many countries have made, but it's certainly something to think about when you have rapidly rising trajectories in some areas. If we look at the vaccines, the inactivated vaccines really don't have published phase 3 data. The one that we have in India, Bharat Biotech's vaccine, has an unusual adjuvant. So we need to set that vaccine a little bit aside from the other inactivated vaccines. And today there is real world data from Chile that shows that 1 dose of Sinovac vaccine gives about 17% protection, and 2 doses are needed for 67% protection against symptomatic disease and about 80% protection against mortality. So unlike many of the other vaccines, a single dose of one inactivated vaccine is not doing very much. Will that hold for Bharat Biotech? Until we have effectiveness data, it's not possible to say. The protein subunit vaccines are very promising because these are vaccines that can be produced at a very large scale. The Novavax vaccine gives 96% against efficacy against the ancestral version of SARS-CoV-2, 86% against B117, and about 56% in South Africa in people who do not have HIV. This is not a vaccine that has gone to regulators as yet. It is under rolling review with the MHRA in the UK. With mRNA vaccines, we've had lots and lots of good news, a brand new platform where vaccines can be made really quickly, where vaccines seem to work with really great efficacy, and they start to work 10 days after the first dose. These vaccines are now already in wave 2, which is we are trying to make these vaccines against variants, and phase 1 studies with the South African variant B1351 based targets have already begun. And these vaccines are now being evaluated in children. Pfizer has gone down to 12 years of age. Moderna is going down to 2 years of age for these vaccines. When we look at how these vaccines are doing in the real world, we have Israel, which decided to serve as a living laboratory for Pfizer, so paid top dollar and also shared data. And essentially, the vaccines are working really well. They protect against infection, they protect against symptomatic disease, they protect against hospitalizations, they protect against mortality. So everything that you're looking for a vaccine, at least in the short term, is being found in the real world as well. When we look at vectored vaccines, we have the AstraZeneca, we have the Gamaleya vaccine, and we have the single dose Janssen vaccine, which is going to be made by BioE. Now, the AstraZeneca vaccine showed variable efficacy depending on the interval, and the US trial had 76 to 79% efficacy in data that came out a few weeks ago. Now, if we look at real world data with the AstraZeneca vaccine, the UK and Scotland decided that they were going to try to vaccinate everybody with the first dose. This vaccine was actually intended to be a single dose vaccine. So Scotland and UK decided that they were going to give both the Pfizer and the AstraZeneca vaccine as a single dose. And it seems like the AstraZeneca vaccine is working really well. It's 94% efficacy. This applies even in people who are over 80 years of age, where there is 81% protection against hospitalization in the first data that came out from Scotland. From the UK, we have data until March 31st, 2021, and vaccine effectiveness of a single dose against symptomatic disease was about 60%, and against death was about 80%. So these data are accumulating, and different countries have decided to use different dosing intervals, with Canada choosing to go all the way out to 4 months. Both the immunogenicity and the efficacy data seem to support the use of longer intervals. We've also started, once we use vaccines in the real world, to see safety issues. With the Moderna vaccine, anaphylaxis is occurring in 1 in 3 lakh people who receive the vaccine. With Pfizer, it's 1 in 1 lakh. With the AstraZeneca vaccine, we are seeing in Europe thrombosis with thrombocytopenia, 1 in 1 lakh, and about 1 in 250,000 in the UK. The Johnson & Johnson vaccine use has been halted in the US because of thrombosis, and on the 23rd of April, the US CDC's ACIP is going to be reviewing the data for Johnson & Johnson. In India, we have an AEFI committee that reviews data. We have a 3-tiered structure at the district, state, and national level, and we've been getting a trickle of data out that looks at causality assessment. And here you can see the only 3 cases that have been published so far where there has been some relationship with vaccination, 1 with Covishield and 2 with Covaxin. None of them are cases that resemble what has been reported for the AstraZeneca vaccine elsewhere. Moving now to variants. Variants actually emerge all the time, and perhaps this is a dendrogram that shows when variants emerged and the numbers of sequences that you can see in GISAID, which is the global database for these sequences. But I think this tile plot shows more clearly that variants are very, very common. So the big grayish blue block that we see on the left is B117, the UK variant. But you can see that there are a lot of variants that emerge that actually have no functional consequence. The variants of concern that we need to think about are variants that can be not detected by the tests that we have, variants that can escape either drug responses or immune responses, or variants that evolve to become more transmissible. And there are questions now about whether immune pressure leads them to more immune escape versus more transmission. Now, in terms of looking at whether vaccines will work against these variants, a lot of studies have been done, and it seems like the UK variant does have a decreased protection from vaccine, but because the immune response is so good, you can't really see that signal. And one way of thinking about it is the real world data that I just shared with you came out at a time when practically all of the virus circulating in the UK is B117. So it's working against that quite well. Now, B1351, the South African variant, is the one that is of great concern. And these have what is called the EEK mutation, or E484K. Both P1 from Brazil and the South African variants have this mutation. And vaccine trials done in South Africa have shown that there is lower efficacy of the Novavax, Janssen, and AstraZeneca vaccines. Even though there is lower efficacy with Novavax and with Janssen, with AstraZeneca, it was said that this vaccine does not protect at all, but there were no severe cases for which a comparison of performance could really be done for that vaccine. The Indian variant is B1617, the famous double mutant. It has E484Q and L452R, both of which may be responsible for immune escape. Both of these have arisen before, and it does not have the mutation N501Y, which seems to be responsible for increased transmissibility. What are the biological properties of this variant? This is something for which we are still waiting to find out. This is a comparison of the main variants of interest and variants of concern. And there is at the moment very little clarity on why and how important the Indian variant will be in terms of spread of disease, as well as how well vaccines will work against this variant. Now, I'm not going to walk you through a lot more information on variants. I want to leave some time for discussion, so I will leave you with the top 10 myths. The first one is the vaccines aren't safe because they were developed quickly. They were actually developed based on work that was already happening on MERS and SARS coronavirus vaccines. We knew the spike structure. That's why it was possible to design a vaccine really, really quickly. And in terms of the platforms that are in use, these platforms had been evaluated in thousands of participants before we put the SARS-CoV-2 vaccines into humans. We can see now that with the pharmacovigilance programs that operate in Western countries, even if you have rare side effects, we can actually detect them. The vaccines can lead to long-term effects. This is something that we don't know, but it does seem to be unlikely. Most of the side effects that we are seeing with these vaccines are effects that are manifesting in a very short time frame after vaccination, and we are continuing to track safety. The vaccines are all non-replicating vaccines so far. There are live attenuated vaccines in development, but they haven't started to be used yet. So there is no question of these vaccines going into latency. or causing disease. Now, if we had an inadequately inactivated or inadequately attenuated vaccine, it would be possible to get COVID-19 from the vaccines. So far, that does not seem to be the case. People say that COVID-19 infection itself should protect, so I don't need to get vaccinated. Well, one of the things that we've seen is that if you develop asymptomatic infection, you actually make a relatively low antibody response. You do make T-cell responses, but whether T-cell responses alone would be enough to protect, we don't know yet. The next two questions about underlying conditions and suppressed immune systems are questions that are of importance. But I think for diabetes and hypertension, which are the common comorbidities, it's very important that people get vaccinated because their risk of severe disease is greater. And for immunosuppression, certain kinds of immunosuppression you can think about being able to alter the treatment with immunosuppressants in order to be able to vaccinate. But people with suppressed immune systems will make some immune response with these inactivated equivalent vaccines. With mRNA and DNA vaccines, there is a worry about integration and alteration of the DNA. Well, for the mRNA vaccines, they never go inside the nucleus. They just deliver the message in a single cycle. So far, our self-amplifying mRNA vaccines have not started to be made, but even there, they would stay within the cytoplasm. There was a lot of information circulating earlier about a resemblance between SARS-CoV-2 spike protein and placental proteins. And the argument was made that this could lead to abortions or could lead to the making of potential antibodies that could result in infertility. This was really cherry picking because if you look at gene sequences and you look at a very short sequence with almost any two proteins, you can pick out sequences that are similar. And that was pretty much what was being done. So both for infertility and pregnancy and lactation, the recommendations, these don't hold true. And the CDC has now modified its recommendations to support vaccination of pregnant and lactating women. Obviously, the one thing to remember is that no vaccines are perfect and you're not going to be protected 100%. And wearing masks and practicing social distancing is very much dependent on how much virus is circulating in the community that you interact with, whether you are vaccinated or not. So I will essentially end with saying that vaccines are working. We made vaccines in 10 months. If the variants do go towards immune escape, we can actually make vaccines much faster. We are already in wave 2 of vaccines. We can move rapidly to wave 3. We need to get people vaccinated as quickly as possible, even as we continue with non-pharmaceutical interventions, because it is the viral load of the whole world that really matters for control of the disease. So I'll stop there and I'd be happy to take questions for the next 4 minutes.
Thank you, ma'am. I know you have a hard stop, so if folks can either, so there's a question here in the chat box, which is, are antibody titers a reliable index of the need for vaccination? Which I sort of think you answered.
Yeah, so actually, we don't have a correlate of protection at the moment. There are attempts to establish a correlate, and these are looking at most likely it will wind up being neutralizing antibodies being used above a certain cutoff, but at the moment that has not been validated.
A couple of more questions. I'll read them out both together. The first one is there data on efficacy in Indian populations on these vaccines? And the second is on the concept of immune escape, is it related to the lack of protection from infection or disease severity?
So the first one, we have an interim analysis from Covaxin that shows 81% protection. We have no protection data from Covishield. It is possible to do vaccine effectiveness studies, and I hope that they will be done. In terms of immune escape, it's really around the virus evolving away from being neutralized by antibodies that are produced in response to infection or vaccination. So the new variants should not be neutralized by the antibodies that were produced in response to an older infection or an older version of the vaccine. Now, what we've seen actually in real life is not too bad, because we are seeing that most of the variants continue to have neutralization, even though it is neutralization requiring a lot more antibody. It seems like from the evaluations that have been done by WHO, by the National Institutes of Health and others, that we actually need pretty little antibody to be able to neutralize the virus, sufficient for it to protect against severe disease and mortality. There is protection that happens from infection for the mRNA vaccines, and to some extent for the vectored vaccines, but important to remember that this is a mucosal infection. We should not expect protection from infection. We should be looking for protection from symptomatic disease and mortality.
Thank you. And there's a bunch more questions that I know you have to go. I'll call one out that I think is on a lot of our minds, which is how serious is the immune escape with the Indian variant B1617?
We have no clue. So in order to do that, you actually need to establish a battery of variants, get sera from people who were infected with the old version, were vaccinated with the different vaccines, and then check for neutralization. To my knowledge, this has not been done. I'm trying to encourage people to do it. So hopefully in a month or so, we should know. But actually, the UK has this panel, and now that they have 100 cases of the variant, I'm pretty sure they will produce results before we do.
No, thank you so much, ma'am. I know there's a bunch more questions. We'll try and get answers from you for these questions later and then post them in our WhatsApp groups. I know you have a hard stop, so thank you so much. We're very grateful to have you.
Please email them to me. I'd be happy to answer any questions.